From Microbe TV, this is Immune, episode number 101, recorded on January 13th, 2026. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, everybody. Great to see you. Hope you can get a chance to see that Immune 100 that we did. That was so fun being in person, everybody together right before the end of the year.
That was a lot of fun, yes. Very festive.
Yep.
Also joining us from Madison, New Jersey, Brian Barker.
Hi, great to be here. It was great to see everyone in person. And now it's great to have a new year of new immunology.
And from Cleveland, Ohio, Steph Langle.
Hi, yes. I like New Year, New Immunology. That's a good
New Year, New Immunology.
Tagline. Yes. Yep. Good to see you all.
Happy New Year, everyone. Hope you have a good year. Someone said it has to be better than last year, but already it's worse.
Yeah.
Well,
it's worse.
Be careful.
What? Careful of what?
Don't say it can't get worse.
No, it's already worse.
I know.
Someone said it can't get worse, and it's getting worse.
It's good to be prepared for worse, but it's good to, I always think of it in historical context. We're still better off than we were 200 years ago. So I do like this timeline better than the 1800s.
Yeah, but things are happening that are really bad.
I'm with you.
And I'm just shocked at what's happening.
No, we gotta keep.
It's just the first 2 weeks, and already many horrible things have happened.
The whole vaccine schedule changed.
Oh, talking to pediatricians.
Flipped
vaccine
the
schedule,
nutrition stuff.
The taking over of other countries, killing innocent Americans.
Correct.
I mean, I can't sleep at night. I really cannot sleep because I worry where we're headed, but we have a chance in November to change all this.
And I like to think of it as, okay, taking the worry, putting it into action, what we're doing now, science communication needed more than ever, talking about the actual science, and just keep pushing on.
Do our little part.
Yeah.
Yes, we are doing. We give you science. We give you truthful science. We talk about papers. You know, you do science. You do experiments. You have a hypothesis. You do experiments, and eventually you publish it. And we talk about the papers. We tell you what the papers mean. We don't lie about them. You know, someone of very high position mischaracterized a Cochrane study on the effectiveness of influenza vaccine.
The Cochrane collaboration, which is kind of one of the ultimate arbiters of vaccine safety and clinical data, has done an extensive meta-review of the flu vaccine. And they found that there is no evidence that the flu vaccine prevents serious disease or that it prevents hospitalizations or death in children.
Yep.
And used it to say probably kids should not get influenza vaccine.
How would it be?
Fewer people will get the flu vaccine.
Well, that may be, and maybe that's a better thing.
I mean, they just lied about what was in the paper.
Right.
And I cannot get that because I was trained that you see what's in the paper, right?
Yeah.
Oh my gosh. So this is what we give you. We tell you what's in the paper and we talk about, what we think and so forth. But if you like that, please support us. We need your help to do this. We have other podcasts as well where we're trying to do it. So go to microbe.tv/contribute. And, yesterday we got a box here at the incubator full of hand-knit caps that a listener made for us to use to raise some money. So we're going to be putting them on our website. There are just like about 40 of them for all different sizes, even little tiny ones for toddlers. They're so cute. And they all have a double-stranded DNA on them.
That is cool.
They're very cool, all different colors. So this person who wants to remain anonymous did this 2 years in a row now. Really a skilled knitter.
That's amazing. I didn't know where you were going with that when you said that a box was sent to the incubator.
Yeah, right.
I'm like, it could go either way. I don't know what you're about to say. Like the good way.
All these caps are sitting on the table behind. It's just so cool.
Oh, I love
that this was done. So we look for that if you want a cap and then you want to have science on your cap. It'd be very cool. Okay, Cindy is going to start off 2026 with a snippet.
Yeah, I picked a snippet that, so I love CAR T cells. We've done them a couple different times on the show, and they're just such an incredible innovation, and exploiting all the things that we learn about the basic biology of how B cells and T cells work, and then taking that to treat cancer, I think is just the coolest thing. And I found this paper and I just thought it was cool because it is from Immunity and it is just out. It's a 2026, fresh off the press. Its press date is today, January 13th. It was actually out a couple days ago, but it is like paper number 1 of the new year in the journal Immunity. The title is IL-9 signaling redirects CAR T cell fate toward CD8 memory and CD4 cycling states, enhancing anti-tumor efficacy. It's from Carl June's lab, and I got to look at if it's got more than one first author.
Looks like no.
Okay, so the first author is Sofia Castelli. So she has first authorship on this. I was interested in this because, one, I think it's cool to find out what new and interesting things they're doing to change up CAR T cells to make them work better and more effectively, and seek out tumor cells more effectively rather than harming healthy cells as well. And it has IL-9. And I was like, IL-9? Huh.
I was like, do you like IL-9 or you don't know?
I didn't really know what IL-9 did. So I was like, I can learn something.
Yeah. I for some reason, I thought that IL-9 was not at all doing these types of things. And so that to me was the most interesting part, is I was like, oh, that's not what I thought IL-9 did. Cool.
I'm like, I know IL-8 and IL-10.
Yeah.
I can tell you about those two.
There's like 30 some ILs and we know what a lot of them do, but IL-9 was one that I wasn't really sure what it did. So as an introduction first, CAR T cell therapy has just been amazing for hematologic cancers. So B cell lymphomas, T cell lymphomas, things like that, things that are in the blood. You can what you do is you take out a patient's T cells, you genetically engineer them to express a synthetic receptor that has a part of an antibody that will recognize something specific on the surface of the target cell. And it is connected to the hardware that you need to activate a T cell. And you put these completely synthetic, they never occur in nature, receptors into these T cells that you took out of the patient. And then you put them back into the patient and those T cells go find the tumor cells and kill them. Beautiful. So cool. Works great for lymphoma. The problem, and it would be fantastic to use for lots of different tumors. There's a couple of problems. One is that it's sometimes hard to find a target that you can wipe out cells and not cause a lot of problem. So the reason why it works so well, for example, for B-cell lymphoma is you can, for the most part, wipe out all your B cells, healthy and tumor, and still survive. Because you can get immunoglobulin injections, they can take antibodies from you, inject them into me, and we can survive. If you start trying to find targets on cells that are shared between the tumor cell and the healthy cell, and you start wiping out, some random fibroblast, that becomes a serious problem. So there's a huge press to try and find, are there unique targets that you can find that are upregulated on a cancer cell that are not present on a healthy cell? But that's not what this particular paper is about. There's a couple of other problems. The second problem is that if you try to go to solid tumors, so hematologic tumors are liquid tumors, you can have lymphoma, so you can get a mass in a lymph node, but generally it's pretty easy to get to those cells. Most cancers are actually solid tumors. So it's a ball of cells that grow and grow, and it's hard for the immune cells to get into the tumor. And if they do get in the tumor, they get shut down right away by a lot of mechanisms that the cancer cells use to quiet the immune system so they can evade the immune detection. So unfortunately, greater than 90% of tumors are actually solid tumors. So it would be great if we can figure out ways to make these CAR T cells work in solid tumors. And so the idea is you put in the CAR T cells, but then they go into the tumor and they get shut down right away. So if there are ways to reinvigorate or keep these CAR T cells going, that'd be great. And so this group has tried multiple different approaches, as have many other groups, to stick cytokines in with the CAR T cells to see if some sort of cytokine signal could keep them going. And the cytokine that they used in this one is IL-9. And so IL-9 is also known as P40 and also mast cell growth enhancing activity cytokine, which I thought, that's interesting. I didn't really know that IL-9 was a cytokine for mast cells, but it is.
Yeah. So I think that to me is what's really interesting about this paper. Because when I thought of IL-9, I was like, I think that's a mast cell one, but that was the sum total of all my knowledge.
Yeah. I was thinking Th2.
Yeah.
And so I'm like,
and so they talk a little bit at the beginning about how this is not normally found. The receptor for this is not normally found on T cells. And so in some ways, like, yay, we're right. It's not normally a T cell stimulating cytokine. And so I'm just like super excited that they decided in the first place to even try.
I know. So the idea was, what if we put this receptor for IL-9 into the CAR T cells, endowing them with the ability to now respond to IL-9? Does that help? I don't know why they thought that would provide the right signal for those cells. I think there's a lot more known about IL-9 and maybe listeners or experts in IL-9. But generally, what I knew sort of was that it's a common gamma chain receptor. So what does that mean? So have you ever heard of severe combined immunodeficiency? So X-linked SCID, one of the mutations is in the common gamma chain. Basically what happens is knocks out that chain of the receptor, and that signaling component is part of a bunch of different cytokine receptors, including IL-2, IL-4, IL-7, IL-9, IL-15, and IL-21. So you knock out the common gamma chain, you knock out all of those, and it is a severe combined immunodeficiency because basically those patients have no T cells, no B cells, and no NK cells. So it wipes out a lot of things. But the idea here is that they're going to put in the IL-9 receptor, and though T cells already have the common gamma receptor, so they're able to signal through this IL-9. So the cytokine receptors are basically at least two chains, one that provides the ligand specificity and one that provides the signal. So in this case, the common gamma chain is the signaling one. The IL-9 specific receptor provides the specificity for the cytokine. And there's another interesting piece that they mentioned in the introduction and that I kind of knew about, but I didn't really look at it in detail, is orthogonal cytokine receptors. So this is a really interesting concept. So they mutate the receptor so that only binds a mutated form of the cytokine that they want. So you can engineer the system.
They mutate the gene.
Right. Sorry. They mutate the gene so that the protein is a mutant version of the protein.
It's an altered version.
Altered version of the protein.
Proteins are not mutated.
Okay, an altered version of the protein. On Immune, we're not as astute as we are on TWIV. So the altered version strongly about this. You're right, and I'm not being precise. So thank you for the correction.
Start the year out.
So this orthogonal receptor can only bind this altered version of the cytokine. So they had used this before and they showed it worked actually. So if they put an orthogonal IL-9 receptor in and they use the altered version of the IL-9 cytokine, they could drive activation of the T cell, the CAR T cells, and it worked really well, helped them eliminate the tumors. And this is great, but there was some toxicity involved in that, and it's a very manipulated system. So they want to know, this paper, could they actually use the native receptor? Do you need this mutated version? And they use a pretty intense model of pancreatic ductal adenocarcinoma, so PDAC. So if you know tumor models, PDAC model. And they want to co-express the CAR and this IL-9 receptor in the CAR T cells, deliver them, and then see if they work. So they do a lot of background, a lot of the first couple of figures. I really like the data. They were very clear in this paper. The paper was really clearly written. But basically, they show that if they put the IL-9 receptor in, it signals, it activates STATs, which we know are the transcription factors downstream of the common gamma chain. The CAR T cells that have have the IL-9 receptor have more efficacy in vitro, so they kill tumor cells. They secrete more cytokine. And if they implant the tumor in the mouse model and deliver IL-9 by adenovirus, so they inject the adenovirus vector into the tumor that delivers the cytokine locally, and then they give the CAR T cells that have the IL-9 receptor, they got a complete remission out of 5 out of 10 of the mice. And it didn't happen if they had the CAR T alone or the CAR T with the IL-9 receptor, but no delivery of IL-9 cytokine, and the IL-9 cytokine alone didn't work. So there was something really cool about putting the receptor into the CAR T cells and delivering the IL-9 that endowed them with this more effective ability to kill tumor cells.
And the other thing that's really cool about that is that, one potential issue with a CAR T cell is a potential off-target effect of not attacking the tumor. And so if you have to deliver the IL-9 just to the solid tumor site, then it's sort of giving you this additional control of we're only going to have super active CAR T cells in the tumor site and not elsewhere.
Yeah. And that's the idea, this on target off tumor. So that means the CAR T cells are attacking the right target, but it's on the wrong cells. It's on healthy cells, and you want to try to avoid that. So again, so if you have a solid tumor and you can deliver the IL-9, you're going to activate those CAR T cells primarily in that tumor. So that's really cool. I also liked in this paper that they always went and kind of did a human comparison as well. So they did a xenograft of human PDAC. So the other one was a mouse model. Human PDAC cells in multi-deficient mouse. And then they were also able to show that they could do 10 out of 10 mice went into remission. They killed their tumors. And in fact, if you re-challenged them, they were still resistant. So these CAR T cells that have got this IL-9 receptor, the endogenous version of the IL-9 receptor can respond to the cytokine. There was a bunch of interesting things in the paper where mouse cytokine versus human cytokine, and could they bind to the receptor and everything. I'm not going to go into the details of that, but you can check out the paper if you're really interested. One of the things they showed was these cells induced potent production. These CAR T cells induced potent production of cytokines like interferon gamma, TNF, and IL-10. And one of the things they looked at in detail was any of this effect, that their better efficacy due to IL-10. And so they used IL-10 blocking antibodies, and they did not. They determined it was totally independent of IL-10. But the big question is okay, so they work better, but why? Why do they work better? So they did a whole bunch of different things. They showed in vitro that the CAR T cells expanded more, so there were more of them. They persisted longer, so they were more resistant to cell death. They definitely infiltrated into tumors when they were injected in vivo. They go into a lot about, that was in vivo. They go into a lot about generating some early memory cells rather than going towards exhaustion. And they go into great detail about this because they do some studies in vitro where they stimulate the cells with targets, and then they do RNA-seq, single cell RNA-seq, and so forth. But it is a lot of data, but the bottom line is that the trajectory of the CAR T cells, if they have the IL-9 and they respond to the IL-9, is to go towards an early memory, but not get towards an exhausted phenotype. So if we go back to what I was saying at the beginning, one of the key things that happens is if the CAR T cells or any T cell gets into a tumor, it gets shut down really easily. And one of those things is it can be driven towards exhaustion, so upregulation of exhaustion markers, or it can be shut down by things like PD-L1 or TIM3 ligand, which will shut down the T cell responses. So it's both of those things. And they show that really these T cells take this trajectory, and they do trajectory analysis and RNA velocity analysis to look, and they show that that when you give them IL-9, they go down a different path, or they go down the path less efficiently. So they're more precursor oriented rather than exhausted, overactive T cells. They also showed a couple of other things that were interesting. With IL-9 stimulation, they also downregulated PD-1 and TIM3. So I just mentioned those ligands. When the cancer cells will upregulate the ligands for those, and when they're engaged, they are negative signals to the T cell to shut them down. So IL-9 actually lowers the level of those receptors on the CAR T cells, so they're less shut down by those signals.
All of this to me was fascinating, having spent some time thinking about CD8 memory development and cytokines, because all of this is really great T cell biology, this really interesting T cell phenotype, and it's happening with a cytokine that is not normally on T cells.
Yep.
And so thinking about, what's going on in terms of evolution, is this just a similar type of response with similar sort of stats that, a similar stat profile gets turned on with some other as yet unknown signal? Is this something that we could somehow try to manipulate in other types of T cell responses to get a T cell to do the things we want to do? I just found this sort of fascinating and was thinking a lot about, both why does this work downstream of a receptor that's not normally on T cells, but also how could this knowledge sort of influence our general T cell biology uses?
Yeah, I was trying to dig through this really quickly because I remember something about, they did show one of the stats that's not normally activated in the T cell very efficiently is activated by this IL-9, and I don't remember which one it was. Sorry.
Yeah, I don't really remember
find it right now.
I was looking. I can look through.
Yeah, because the ones that are normally activated are, what did I write down?
So I see 4 here.
Stat 4.
Maybe it's stat 4. I think it is stat 4.
Well, stat 4 was the canonical one.
Well, normally IL-9 activates stat 1, 3, and 5. And I think it activated 4 in those CAR T cells, which is interesting because it's not normally activated by that receptor. And they did talk about that a little bit in the discussion, why that might be.
Oh, I see. Yeah, I see what you're saying. Like non-canonical to IL-9, canonical for like IL-12. Yeah.
Yeah, when I think of common gamma chain, I think of 135.
Yeah, exactly. Yeah. So it's something unique about putting that receptor into the CAR T cells that gives them resistance to being shut down when they get in the tumor. They actually get in the tumor better. They keep their CAR expression. So sometimes CAR gets downregulated. They seem to be producing these early memory cells and proliferating more. Not going down an exhaustion phenotype. And then maybe that's all because there's some fine difference in how the receptor is mediating the signal in these cells that is normally not active in.
It's too bad they have to inject it because, these tumors do metastasize.
They do.
Can't inject all the mets.
So I think the idea is if you engage the CAR and you activate it and you provide it with these signals, they're not all necessarily going to stay in the tumor, and they can travel out and find the mets. I think that's the advantage of using a living therapy like a CAR T versus some other types of therapies, is that those cells can then migrate around the body and find the mets and eliminate them. And the smaller it is, the easier it is for the CAR T cells to eliminate them, because once it gets bigger, it's harder to get inside once you have the solid tumor, and then the tumor microenvironment changes and shuts down T cells.
They could test that in their mouse model.
They
could have a metastatic model.
Yep.
So how do they deliver the IL-9 to the tumor? Is there vectored?
So, in their model, it's in an adenovirus.
Okay.
Yep. And they provide all the data that shows that the adenovirals or the tumor cells express the cytokine, and that it's higher in the tumor. And they could differentiate between the mouse IL-9 and the human IL-9 when they were delivering with the vector, and show that it's in the tumor and not in the serum. So they had all those controls in there.
Well, since they already have a tumor-specific antigen in the CAR T, the chimeric antigen, maybe they could use that to target a virus producing IL-9 to the tumors, metastatic tumors, even just do IV or something, because that's been shown in other situations to work, If it doesn't work, just as you say, the CAR T cells getting activated in some way, they could try that. would be good.
Yep.
And so do you see, future work trying to understand, if that STAT4 activation, like, is it truly downstream of IL-9 or is it the secondary cytokine?
Yeah, so they've mentioned a little bit about that. They said, although STAT4 deletion revealed functional effects on cytokine production, so the STAT4 was important for the function, compensatory activation of other STAT family members, such as STAT1 or STAT3, may have masked the full extent of STAT4-dependent effects. So it seems like they're really interested in looking at what the individual STAT protein products are doing in the presence of the IL-9. They're not really sure. They know they're important, but they don't really know what they're doing. Yeah. So I guess one could just delete them and then see like gene expression profiles and things to see how each stat is contributing to the overall outcome of the CAR T cell activation. Yeah.
Or then I guess you could block the canonical, like IL-12, the canonical receptor for, that signals through STAT4, and see if that would be a secondary cytokine, that if these IL-9 induced effects are causing secretion of that, which further would upregulate that STAT4 signal. It's very interesting. Because I guess this idea that, like, what are other cytokines receptors that you could use to signal this desired effect that aren't canonical? That's really intriguing.
Yeah. Most of the approaches have used like IL-2 or IL-15, because those are, traditional T cell proliferation factors and things. But I guess that is not endowing them with in this case, the ability to evade going down an exhaustion. Yeah. So if you just keep proliferating and you keep stimulating, you're going to end up going down the exhaustion pathway.
That's fair.
Yeah. IL-2 has a pretty well-defined role in pushing to exhaustion.
It's also kind of toxic to deliver.
Yeah, exactly.
Yeah,
so. IL-2 is I think of as having a very Goldilocks type of profile in terms of, if you're just right at the just right time, it's awesome, but it can also be not awesome.
Yeah.
It's fair.
Yep.
Very interesting.
Yep. Thank you, Cindy.
Yeah, sure.
All right, I found this paper last year, and I was waiting for an opportunity on immune. This is about tattoos. PNAS. Tattoo ink induces inflammation in the draining lymph node and alters the immune response to vaccination. First author is Arianna Capuchetti, Italiana.
I figured you'd like pronouncing her name.
And then the last author is Santiago Gonzalez. Oh, there's so many. There are a bunch of, this is from a variety of institutions, many of which are in Italy.
Don't forget they do have 2 first authors in this one.
Yeah, I couldn't find that. It's all the way at the end.
Giuliana
Falivene. I'm going to say all the Italian ones. Chiara Pizzichetti. Luca Mazzucchelli. Wow. Andrea Raimondi. So that's cool because Raimondi is my middle name. Nobody knows this.
Oh, is that right?
Huh. Nobody knows. Well, a few people
now
know,
they do.
Yeah. It's Vincent R. And I never tell anyone because it was the name of my maternal, my mother's father.
Oh, wow.
Raimondi was his name. And it's the tradition to give middle names like that in Italian.
Is that the Italian equivalent of Raymond?
Not really, no.
No?
No. So people say, can I use Raymond? I said, no, it's not the same. It's Raimondi. But it's funny. My mother, when she was young, she wanted to be cool, so she would spell it with a Y. I have some of her old books from the 30s and 40s. Raymondi instead of I. And I asked her, I said, she said, I was just young and silly.
Just wanted to play around with it.
Wanted to play around with it. What other cool ones here? Anyway, tattooing is really popular. They say the first sentence, tattooing has become widespread for young people. 1 in 5 people globally have tattoos. And the U.S. has the highest prevalence. 30% of the U.S. population is tattooed. Now, I don't have any because when I was young, it was not popular. However, I always wanted a little virus tattoo on my hand, but no, after reading this paper, no way.
Out the window now.
Oh my gosh, this is so scary.
Although, as we'll find out, maybe your flu vaccine, maybe there's
when you find out what's happening, you got to make your own decision. So, tattooing, they take needles with ink in them and they put them into the dermal layer, and you can use different types of pigments. And these pigments are not soluble in your body, so they stay there. And these pigments are, they're a little scary. They're made up of pigment binders, solvents, and additive. Now, carbon black is the primary pigment used in black tattoos, right? And they have been, the carbon there has been well studied and it's safe. But the colored tattoos, they often have industrial organic pigments that are made to be put in plastics and varnishes and paints, not in the human body.
Man.
And this is a little dicey.
But those color tattoos, have you seen? They are cool.
Color tattoos are cool. Yeah, a lot of people have them.
Yeah. But you're kind of just playing with fire there, I guess.
We actually did a paper a couple of years ago.
The macrophage one?
Yeah.
Yeah. So the macrophages take up the tattoo ink.
And then die.
Yeah, then they die. They're going to see that again here. And mouse studies have been done showing that the tattoo ink often accumulates in the lymph nodes that drain the tattoo site. And this is also seen in people. So, for example, women who have had mastectomies, they take out their lymph nodes. If they've had a tattoo, they look and they find the dye in those lymph nodes as well. So this paper, they're going to revisit this.
It's not a surprise that much. I mean, it's a surprise that they're long term, but I guess if you can't get rid of it, it makes sense. But if you inject dyes into a mouse or whatever, it goes into the lymph node, right? I mean, pretty much anything you put in the skin will go into the lymph node.
Yeah, it's the persistence that's really the...
Yeah, that's the thing here, because these dyes are insoluble, I guess. That's part of the problem. Yeah, and they just stay. Of course, if they degraded, then the tattoo would fade, so that would defeat the whole purpose. So.
Yep.
Anyway, so they're studying this in mice and a little bit of human results as well. So they're looking at the most popular colors: black, red, and green. Okay, and they buy these from a producer of tattoo inks, and then they have a mouse model where they tattoo a 25 square millimeter part of the foot pad of the mouse. Now, these are now cool mice, because they have tats, but they're not designs. They're just rectangles.
I was wondering how small that is, but it covers a fair amount of the poor little mouse's foot.
It does.
It does.
They have pictures of it here.
It kind of looks like a glove, like when they get those ones where it covers the whole arm or the whole sleeve. Neck. And, the sleeve.
Oh my gosh, that's scary. The sleeve. I had a student who got a sleeve tattoo.
Wow.
From the shoulder all the way down to the wrist, completely covered with ink.
Can you imagine what their lymph node looks like?
That's a little bit like with this mouse, I guess.
Oh, gosh. So then they can see by EM that the ink is in the epidermis and the dermis, and it's associated with certain cells that look like keratinocytes, macrophages, and the collagen matrix. And then they surgically expose the hind limb, and they do intravital imaging right after they tattoo, and they can see the ink going through the lymph. They have these nice pictures of that. They go through the popliteal lymphatic vessels, and they reach their peak about 10 minutes after tattooing. And then you see the ink accumulating in the popliteal lymph node about 24 hours post-tattooing. So they have a new term, PT, post-tattooing. Okay, PT, for all of these inks. And in fact, in two inks, the black and red, they also find them in the lumbar lymph nodes. So it's spreading even farther. So this is ink that hasn't remained in the skin, obviously, right? They call it unretained ink. At 24 hours, there's the lymph nodes, they have ink in them, right? And they also get bigger. The lymph nodes are getting bigger.
Which is suggestive that there's some sort of inflammatory reaction happening.
Yeah.
Yep.
Right. And they also have some human biopsies, lymph node biopsies from patients who had a tattoo, and they biopsied the draining lymph node, and they see this several months after tattooing that the ink is still in the lymph node. That's in humans. Okay. So what cells are capturing the ink so they stain for a couple of different kinds of phagocytic populations, subcapsular sinus macrophages, medullary macrophages, and dendritic cells. They look at 24 hours and 2 months PT, and the medullary macrophages are the primary cell type that are taking up the ink. The dendritic cells can take up a little, but it's mainly the MMs.
I would have thought that the subcapsular sinus macrophages would do more.
Yeah, why is that?
Because the lymph flows into the lymph node.
Yeah.
And then it goes around the subcapsular sinus and then gets into the main part of the lymph node. And the subcapsular sinus macrophages are the ones that capture things out of the lymph and bring them into the lymph node. But maybe it's just temporary. Maybe they just suck it up and spit it back out on the other side so fast that they can't capture that.
So what's happening here is the ink is flowing into the node and then it's being captured. Is that how it's working? Yeah.
Do you think it's are medullary macrophages? Do they have a higher rate of phagocytosis?
I think so.
So maybe it's that's interesting about the timing. Maybe they can't capture
the very very
the capsular ones, and then these are just what they can capture, and also could be a rate. Maybe they pick it up,
and it just ends up there. It's going to end up in those macrophages. Yeah.
Right.
They also embed these lymph nodes and look at them under transmission EM, and they can see that the ink particles are associated with phagocytic cells, which looks like macrophages. They can see vacuoles within these cells with the ink, and they can see the increase in the size of the cells and the amount of ink associated with macrophages. And they also do this with 2 biopsies from human lymph node biopsies from tattooed patients, and they could see ink associated with macrophages. So is this bad for the macrophage? They measure the total number of cells in the popliteal lymph node of these tattooed mice by fax during the first 5 days. They see an increase in the total number of macrophages in first 6 hours PT, and then it goes down at 12 and 24 hours. And interestingly, that's just for the red and black ink. Green ink, they remain decrease. They don't decrease until 120 hours PT. So there are obviously some differences in these dyes, right? Which is not good. So they think the disappearance of these macrophages has something to do with the ink particles. These cells undergo morphological changes. It looks like something like cell death. So what they do is they isolate mesenchymal stem cells from the bone marrow of mice. They differentiate them into macrophages, and then they add ink. They don't tattoo the macrophages. They just add ink to the cultures, and they see they're taken up. The ink is taken up by these macrophages, and then they seem to be undergoing necrosis. They can stain them with propydium iodide, which is a marker of necrosis. And so there's an increase in the number of PI staining cells 12 hours after adding the ink with the red and the black inks, but not with the green ink. No toxic effects of the green ink. I guess if you got to have a color, you might as well use green, right?
Green ink.
Yeah. Well,
green and black.
I think it's sort of interesting in the in vivo part that, they see this decrease, but of course before that they see the increase in macrophages in the draining lymph node, again sort of suggesting that there's some inflammatory process going on.
Right.
I think it would be really interesting to look at these cells that were just directly treated with ink and look at changes in cytokine production or transcription or something like that and see what kind of inflammatory changes were happening just after that direct ink exposure.
Right. The inks also induce apoptosis, which they can measure by annexin staining, and in the first 48 hours after adding the ink.
One has to imagine that some of these things are always happening, right? Because you're going to get crap in your skin. You're going to get, abrasions and things that will have bugs, will not have bugs. Bugs get into the lymph node. Lots of things flow into the lymph node. I think the difference here is that you can't get rid of that dye. It just stays, right? Sure. So it's just got to be captured and put somewhere. And the question is the impact that it has, is it long lasting or is it just temporary?
Yeah. Well, we really do need long-term studies, right? But it was limited here in mice. But I would guess if you looked at mice for their lifetimes, you'd still find the ink in there. It's only a couple of years, right? They find the same things with human macrophages differentiated from PBMCs. They have similar things. They add ink, it gets taken up, and it causes cell death. And black and green inks, in the case of humans, black and green inks induce apoptosis and red less so. So it's weird that dyes have different effects in mice and humans. Okay, what about inflammation? So we've talked about inflammation. So they say maybe the ink is inducing inflammation. So they count the number of cells after tattooing with black ink. This is back in the mice now. They see an increase in the lymph node at 48 hours. And that keeps increasing up to 240 hours post-tattooing. And they also see B cells, T cells, and NK cells, as well as DCs going up after tattooing. They collect the lymph from the popliteal lymph node during the first 240 hours after tattooing. They measure cytokines, inflammatory cytokines. And they see them. They have IL-6, CXCL1, CCL2, CCL3. They peak at 6 to 12 hours post-tattooing and then go back down. They also identify a second group of molecules, which include IL-1 alpha, which have longer-term expression. They remain elevated for 240 hours post-tattooing. So this is in the lymph. Now they look in the blood. So they say, is this getting into the blood? But yeah, that's what cytokines like to do, right? They go everywhere. So they measure the mediators in the blood at 5 days and 2 months after tattooing. So they also see the early peak of these inflammatory cytokines and chemokines that we've measured. And again, they're transiently elevated. They go back down again. But IL-1 alpha remains elevated in the blood for 2 months.
Yeah. It was interesting that you saw an elevation up until 120 days, but it was pretty variable. You had some mice that really didn't have much IL-1 alpha and some that had very high. But then you're right. Once you get later on, it's pretty consistently different.
Right.
Yeah.
They also measured B cells, T cells, NK cells, and DCs 2 months PT with black ink, and they see those increasing as well.
One thing I was wondering about is when you get a tattoo, usually keep it covered for a certain amount of time because it's sensitive and it's also susceptible to infection, right? But you're tattooing the bottom of a mouse's foot that's walking around in a cage.
Right.
So I wonder if some of what they see might be secondary to bacteria that might get in and cause some inflammation. I didn't look that closely at the details of their methods to see if they covered the foot or did anything to prevent infection.
Well, what they did is they poked them without any dye.
That's
true. They did that with PBS, which
they have PBS and they have non-tattoo. And it's not always super clear in the bar graphs. I'm kind of assuming the non-tattoo is basically 0 and everything else is how much above non-tattoo because there isn't a clear bar for those.
Yeah.
And if that is the case, then the PBS did induce some inflammation.
Right.
But the ink was much more.
Well, the poking of the skin is going to do inflammation, right? Yeah. Big time.
But I also wonder, though, because you're going to get into some effects that they have on vaccines, but they could also have effects on the immune response to bacteria that might be taken up too, especially if it's affecting the cells in the local tissue.
Yeah,
but
they didn't do like a shoulder. Right, we didn't get a comparison between a different site. That
it could have shaved the mice and tattooed a different spot that was not walking around in their own feces.
One of the things that
I would wonder about tail.
Yeah,
because with shoulder, then you have it does the shaving influence.
You could, I guess, shave a you could, just shave an animal and see if it matters. But the other thing, don't people tattoo the ears sometimes if they don't want to do ear tags?
And we also used to tattoo tail.
Oh, you did? Okay.
And so that's why I was thinking about that.
Yeah. I imagine the ear's not as vascularized, so it probably, I don't know, maybe it wouldn't have such an effect, but
I don't know, but they could certainly just 2-photon it and see everything that's going on in the ear following a tattoo.
That's true.
Because you could just put the ear on a mount and just visualize right straight through it.
Right.
Mm-hmm. Yeah. Well, that's interesting.
And then the last set of experiments, they asked whether tattooing alters the immune response to vaccination. So they do 2 different experiments. The first one, they immunize mice with the Pfizer COVID mRNA vaccine at 2 days and at 2 months after being tattooed with black, red, or green ink. So tattoo, then 2 days and 2 months later, they get immunized. And then they measure antibody responses against the spike receptor binding domain. So all the tattooed group make less antibody against the RBD at 10 days post vaccination. But only animals that got red and green ink had decreased level of IgM at 7 days.
That's interesting.
The reduced IgG was in all groups tattooed 2 months before, so it lasts for 2 months.
Huh.
Right. Then they say, okay, so remember the mRNA vaccines are injected, and then the mRNA is translated, right? And then the mRNA is translated into protein. So they ask whether the ink, the tattoo ink, affects the expression of the coronavirus spike protein. So they immunostain with antibody, and they find that, in fact, in these medullary macrophages from all the tattooed groups, they have reduced levels of spike protein production in the short-term experiments. So what that could be part of the effect on the antibodies, I guess. So then they do another experiment where they immunize 1 foot pad, and then they tattoo 1 foot pad and then immunize the other foot pad, contralateral, and there's no effect on antibody responses. So I guess if you have a shoulder tat, you get immunized in the other one.
Yeah, for mRNA vaccines.
At least for mRNA vaccines.
Yeah.
So then for humans, they differentiate macrophages from PBMCs of 6 donors, and they add different amounts of the ink together with the vaccine, and they co-culture them with B and T cells, and they see that all the inks reduce the anti-spike antibody produced by the B cells at 9 days after adding the dye to these cultures. And the black ink in particular reduced the expression of the spike protein in these cells.
Now, do we know if there's any there should be soluble dyes as well? Do you think there should be a soluble dye control just to show that it's hard to do any control for this because they don't, they're not manufacturing it. They're buying it, right? These are the actual ones that they're using. So you can't get all those other things, the binders and the other things that are in there to see if they alone have any impact, because they could be influencing membrane permeability. They could be influencing protein translation, and it could be the dye, or it could be something in the mixture of the dye. It's hard to say.
Henna tattoos are temporary.
Yeah, they're not in the skin though, right? They're just painted on.
Oh, is that okay? Yeah.
And I think the key to this is that they go into the dermis.
Yeah.
Because then it's going to flow out of the dermis.
I think that's my lack of knowledge of henna tattoos. I didn't really know that.
As far as I know, they are just painted on the skin.
Yeah, right. That's fair.
And stained. Like if you eat blueberries or raspberries or strawberries and it stains your skin. Cranberries, I think, is a good one.
Yeah, no, I think that you're right that a non-soluble dye would probably be useful because that's delivered in exactly the same way.
Yeah.
But you could also imagine that a future study could take the black ink or the green ink or the red ink and actually fractionate it into all of those things and see if their individual components have these same effects.
And people already know that the agents that they put in there do have toxicity. That they did mention that in the introduction.
Yeah.
Like What amazes me is I think about who are the people who refuse vaccines.
Yeah.
And that they all have tattoos. So they're not okay with injecting something in their body that we know is protective and is safe, but they will inject in unregulated dyes into their body and they're okay with it.
Yeah.
I don't get it.
To be fair, because there's a high proportion of individuals who get tattoos. It's a large group. I imagine that, we would have to find the nexus of people who are skeptical. But you know what? I think what really gets us with COVID is that for so many people, like they didn't have to get tattooed to have their job. I think when you get into this, we as scientists understand why this happened, but when you have like work requirements and large institutions telling you to do something, people who are just like I got friends and family, they're just anti people telling them what to do. Yeah. So I imagine if there was organizations, institutions telling them you all have to get tattooed, they might feel, so it might not be like your interpretation of toxicity, because a lot of people still smoke, even though that's like clearly bad, but it's so much context. I only say that just because I got a whole span of
it's also, I go a little bit on a soapbox. It's also these people who are like, big pharma bad, but give me all the supplements that are unregulated, right?
Fair.
Yeah, of course, doesn't make sense.
I think it just is I don't understand.
Hard. And I think it goes back to, like big things that people have no way to change. I actually do. I'm just like going on a soapbox now, but I think actually scientists being a little bit understanding of the anti-pharma, because there are a lot, people have to pay a ton of money that their taxpayer funding taxpayers funded the work that allowed these drugs to happen, but they cannot afford, they will go bankrupt if they have to take them. Yeah. So I do think there's wrong. Like a lot
But that's not science's fault. And that's not actually even pharma's fault.
But if you don't know a scientist personally, which is why I think our podcasts are good, right?
That's insurance fault.
It's hard.
We don't have good health care, and that's because the current admin doesn't want to help people that way.
Well, that is true. That's why I'm like 2026 is me being a bit more showing people that we're different. Like it's a different thing. Like we do need new health care, but like I can't change that as an individual. Right. It's a hard, people do conflate things with.
Yeah. Oh, absolutely.
Well, Obamacare was great for so many people and they would like to get rid of it.
Right.
Well, because they don't know what it is. Because once they realize that they no longer have health care, then they'd be like, wait, you took away my health care. I'm like, yeah, because you didn't want it.
Well, and maybe we should have people on who want to just ask us questions directly. So it can go from like these people to Like, let's maybe have them on the show, right? Because I think maybe we should debate these things on like a podcast format. And I'm not talking people who are antagonistic and want us to like die because you know what I mean? I'm talking people who just like have questions.
I think debating is a bit much, but because I think science, you don't debate science. Science speaks for itself, right?
Yeah. But conversations.
Talking about conversations.
You could go on Vincent's office hours.
I think we should have them on though. But either way, I don't want to distract from your paper.
No, it's fine. It's a good discussion because it's true. Many people say, I'm not putting anything in my body, but they get tattoos and drugs and other things. So I think it's just an excuse for not wanting to be told what to do. Yeah. That's all.
I mean,
I don't have a problem with that.
I mean, what if we could make your vaccine also give you like a nice blue pattern?
Could I put if I were to
combine? Yeah, could I combine the two? Would you take it?
Hey, maybe.
An interesting idea to deliver a vaccine by tattooing.
We did the paper by flossing, right? So why not?
If you're willing to get the tattoo, can I put some antigen in there?
Can I just throw in some flu protein? HA.
Let's not get ideas because then I feel like we're gonna get identification tattoos that also deliver your vaccine at the same time.
Well, we really have to be careful with identification tattoos. That goes down
that's a big fat no.
That's different.
No, that's just bad.
Yeah. The smallpox vaccine works well because of the scarification induces inflammation. And I bet tattooing would also work well because you're poking the skin over and over again, right? Yep.
And delivering antigens to the lymph node.
Yeah. Anyway, they wanted to try a different kind of vaccine because the mRNA vaccines have to be translated into proteins. So they tried an inactivated influenza virus vaccine with the same kind of setup, right? Tattoo and then immunize at 2 days and 2 months. They got different results. The black and red ink increased the amount of IgM at 7 days, and the red group increased anti-influenza IgG at 10 days. And among the mice who got tattooed 2 months before, the ones with red ink had increased level of IgM at 7 days, and black and red elevated IgG at 10 days. So an opposite effect from the mRNA vaccine.
Interesting. So we think maybe because the UV inactivase, like you're just putting the protein in. There's no mRNA translation that has to happen. So maybe the ink blocks the ability of mRNA to be transferred.
Yeah, because remember the antigen, the spike was down in tattooed mice. So that's consistent with that. Whereas, you give them protein and maybe the inflammation is helping in this case, right? The tattoo-based inflammation is helping.
Yeah. I think it just comes down to which antigen presenting cells are presenting the antigen, and they're going to be different ones. If you deliver mRNA vaccine versus if you deliver protein inactivated.
Right. And the sort of mystery ingredients could be stimulating these antigen presenting cells differently. Yeah. And so perhaps an ingredient in green influences macrophages more and an ingredient in red influences some other cell type more. And so I think that actually sort of parsing out some of those individual ingredients and which things are having these effects could give us interesting information on inflammation and antigen presentation.
Right. But I think what the big takeaway is, it is so striking that the response to these two vaccines is actually completely opposite.
Yeah.
Right. And the fact it's really telling you something about the biology that's fundamentally different between an mRNA vaccine and a protein antigen vaccine, which we know is true. Right. But this is actually putting it right in your face saying, this one thing can influence these two vaccines really, really differently. So there's probably a lot of other things that can influence the two types of vaccines or all the different formulations of vaccines. There's so many different formats of vaccines. They all could be influenced differently by different subtle environmental changes.
Yeah. I wonder also what happens with a boost. on that. Does a boost sort of wash out that difference? And so is this another argument for no matter what vaccine you have, you probably should get multiple doses to get rid of some of these individual variations?
Yeah. So they mentioned in the discussion that when you get tattoos, you get about 2.5 milligrams per square centimeter. So they say, wonder people who get these large tattoos, right? Sleeves, or some people who tattoo their entire bodies, right? They're getting many, many milligrams. And so what's the consequence? Probably you're going to have ink in all of your lymph nodes if you do that. So what's the consequence? So they say it's something that hasn't been studied and probably should be. The other thing they say they wonder if the impairment of macrophages could make you susceptible to bacterial or viral infections, right? So that hasn't really been looked at. And then there's some associations between tattooing and different kinds of cancer. So there's an increased risk of lymphomas after tattooing that's been reported. And so that needs to be studied because, inflammation induced by tattooing, it's not good if you have dormant cancer cells that can reawaken some of them. So a lot of things that need to be studied, and probably this needs to be done in humans at some point.
And wasn't there something about like triggering autoimmunity or some weird skin reactions that can happen at tumor sites if you develop some autoimmune disease or something like that? Or did I remember that wrong? I thought I remember seeing something like that in the discussion. I should have made note of it. But yeah, if you lose tolerance to those dyes or whatever is going on with that, it could cause a real problem.
Yeah. They also say that the dyes need to be better regulated. They'd have a lot of components that are not made, solvents, preservatives, additives, non-liquid components, which are meant for industrial uses. So they really need to be regulated.
I think this paper, though, or this paper and this discussion together also highlight some of the reasons why some of this is hard. Because if we think about it, in some of the figures, they looked at some of these dyes in mice and in humans and saw the different colors acted differently in mouse and in human. And so at some level, that testing that we're saying needs to happen with those dyes, we need to think carefully about, because we can already see from these data, the mouse data don't perfectly recapitulate the human data. And I wonder, for humans, if you were to do a study looking at those with tattoos versus those without, there are clearly going to be other variables.
Sure.
And so then do you get a very large population of people and give them a tattoo as part of a clinical trial?
Well, you could identify people who want to.
How many people would you have to have in that trial to do this?
I think you could just enroll them if they're planning on getting a tattoo, right?
You enroll them, right? And then they're going to get one, and you follow them up. But let's look at it this way. If each of you, if someone now emailed you and said, should I get a tattoo, what would you tell them?
It's interesting.
Were you going to say just get the black one?
Yeah, the black one at least is regulated, but it still had an effect. I think.
Yeah, it does kill macrophages.
Kills macrophages. So, I guess it depends on that individual's desire level. If they're just like, maybe if they're ambivalent, I'd be like, then don't. If you're going to kill your macrophages, then commit to it. Don't,
What would you say, Brianne?
I think I would ask them, are you otherwise immunocompromised?
Yeah.
How's your immune function in general? What other kinds of family history do you have?
Well, because that's a good point. What IL-1 receptor alpha being upregulated for many months, what would that do then if you had an autoimmune disease or a flare up, right? Yep. So, yeah.
So what would you say, Cindy?
I don't know. I don't like tattoos.
No, but somebody
to
wants.
each their own, I guess. But I would
let's put it closer to home. If your kid wanted a tattoo,
I don't have any choice. They'll do what they want to do, right? But I would tell them not to. Just like I tell them not to do drugs and not to drink. Probably do it anyway. So
I guess you present them with the evidence.
Yeah.
Yeah.
And
it's hard though, because you make those choices when you're young and you think you're, invulnerable.
But this is mostly mouse data, right?
It
and we
is.
always
That's
say
fair.
you can't directly extrapolate, right?
Well, it is mostly mouse data, except that it was very clear that once you get a tattoo, you can find the dye in your lymph node.
Yes, sure.
And if you incubate macrophages with the dye in vitro, they do die.
Correct.
So yes, it is extrapolation for, effects on vaccine responses and so forth. But we do know that in the skin, the dye kills macrophages and other macrophages go and eat them. And then they take up the dye and then they die. And that's how that's maintained in the skin. We now know that it goes into the lymph node. It gets taken up by macrophages. Those macrophages are inflammatory. The lymph node's inflammatory and maintains that over time. So I don't know. It's injecting something in certainly that is not supposed to be there.
Couldn't you do massive studies? These are going to be retrospective observational studies and just see if people with tattoos have different issues compared to people without.
I think you can, right? I think you can, but I think that then the question becomes, are there other differences?
Yeah, that's the problem. It's going to be confounding factors.
Right.
Yeah.
I mean, as I'm looking at all of this, I'm intrigued and want to talk to some friends who have tattoos, who have gotten them either more recently or earlier in their life, and ask how they feel about it now. About looking at things like this, because I don't know what I would say.
Yeah.
Yeah. In the end, it's somebody's personal choice, because I think here's the difference. We were talking about vaccines before and things. If you choose to inject ink into your skin, that's not going to hurt me. Right? So it will hurt you if this turns out to be true and impairs your immune response or makes you more susceptible to something. But it's not going to hurt me. I think where we can regulate things more is when your choices do actually impact other people, because that's what public health is for. Right?
Yeah.
Oh, look, there's a paper here from 2008. DNA vaccination via tattooing induces stronger humoral and cellular immune responses.
Yeah, no, I know the people who I might know the people who did that paper.
Interesting. Well, if they asked me, I would say a small one, if you must. But
just
I wouldn't
one
do
little virus. It would be okay.
Just a little virus or an antibody or a macrophage.
I mean, isn't that ironic if you got a macrophage tattoo and it's macrophages that are maintaining your macrophage tattoo, and
it must be worth it, honestly, for the story.
Well, I actually found a picture of someone with a macrophage tattoo.
Oh yeah, cool. I'll stick to my
that.
macrophage necklace.
Yeah, there's one or two small tattoos, and I feel like after a certain age, okay, a couple of macrophages may die. I don't know. Small tattoo. I feel like.
So are you going to get one? Are you going to get a B cell or an antibody?
I have two things that I would get, and I don't know if I would get them now. It would be like years, and I gotta for me, if I'm going to make a decision like that, I need to really make sure it's what I want. IgA I would do small IgA.
Yeah, I've seen some that people have that are pretty cute, are pretty good, but I don't know. I also am a wimp.
Yeah, but a small one won't take very long.
Yeah.
Okay.
It's funny.
Well,
this is great.
Steph, we have an email. Can you read that, please?
I can. Let me get back into the notes here. Okay.
Actually, that's the wrong way to ask it. I should say, would you? Because obviously anyone can read it. It's would. Would you read the email, please?
Sure. I'm happy to. Okay. Dear Stephanie, I wanted to drop you a note and express how much I enjoyed your Immune 98 T cells on the brain. I am the senior co-author of that paper with Andrew Wong. I think it's Wong, correct? Maybe somebody looked that up. I believe Andrew Wong. It's always interesting to hear how others view one's work. First, full disclosure, I am the normal subject. And as a neurologist would point out that a lumbar puncture is basically a very low risk procedure in someone who is not anticoagulated. And he notes colonoscopy is part of the routine screening for someone at his age. And so they added the biopsy for this study. Oh. The work, Cindy, you said it one time on an episode. A lot of science sometimes is studying yourself.
On yourself.
On yourself, right?
Yeah.
The work began with our investigation of patients with MS, where we found an inflammatory signal at a single cell resolution, not surprising, in the CSF. And I think what he says, not surprising, is you're going to find a difference in an inflammatory disease at single cell resolution. Jenna, my graduate student, thought we needed to do controls and enlisted the aid of a group of graduate students and postdocs for lumbar punctures. The paper is attached, and to be clear, I made sure that I did not know who was having the LPs, and compensation was $200. I've also attached a paper with under review investigating patients with prodromal Parkinson's disease, where LPs were performed in 15 healthy subjects, and we have extended the number of subjects comparing CSF to gut repertoires. And there's a preprint, and we can link to that. Tomami, which may be the graduate student he was referencing, but that individual,
that's the first author on the Nature paper. I just pulled up the Nature paper.
Thank you.
It is Wong, and yes.
Thank you. Perfect. Okay. Tomami's the first author. After doing a rotation in my lab, Tomami enlisted Andrew to see if we could model the observation in mice. As was pointed out, normal human brain is an oxymoron, and one can't do the labeling experiments that we did in mice to exclude intravascular T cells. I would only add that for me, the work changed the way I viewed the immune system. That is, nature reuses systems for different purposes, and our work suggests that nature uses T cells to sense the microbial environment in the gut, which is dictated by diet and other factors, and transfers the information to the SFO. And if I remember, that's the subfornicular organ.
Subfornical organ.
Yeah. Fornicle. A circumventricular nucleus, not behind the blood-brain barrier, influencing behavior. Similar perhaps to work related to CD4 cell sensing, sodium, thank you, levels in organs. And for those who have not seen a spinal tap, attaches a video made at a Prado Foundation workshop. It was his LP. Best wishes, David Hafler, MD, who is at Yale, and he was a former chair of neurology, and he's a professor of immunobiology. That was a neat email to learn that extra info.
There's a normal human brain is an oxymoron. Huh? That's very interesting.
Yes.
Well, we'll call it control then.
Yes.
All right, it's Immune number 101. You can send your questions and comments to immune@microbe.tv. And if you would like to support these kinds of programs, we'd love your support. Please go to microbe.tv/contribute. Microbe TV is a nonprofit, so in the U.S., you could federally deduct your contributions. Cindy Leifer is at Cornell University. Cindy Leifer on Blue Sky. Thank you, Cindy.
Thank you. I'm wondering, should we have our listeners send in pictures of their science-related tattoos?
Yeah,
maybe we can pick one for.
Oh, that would be awesome.
Anyway, yeah.
Immune@microbe.tv.
Cool. Yeah.
Yeah.
Steph Langle's at Case Western Reserve University. Stephanie Langle on Blue Sky. Thank you, Steph.
Thank you. This is great.
I can see you're going to wait like 50 years till you get your tattoo.
I know. I'm not.
Then you're going to be an older person. I got my first tattoo.
Oh, thanks.
Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Well, here I am, a doddering old guy. I still don't have a tattoo, and I dream of it, and then I come across this paper now. Yeah. Maybe I'll get a temporary one. Maybe I'll get a henna tattoo. How long does a henna tattoo last?
Like 2 weeks or something.
2 weeks.
Yeah, I can get one every 2 weeks.
There you go.
And then it's like a living.
You just need
work
one of those ones
art.
where you lick it and you stick it on,
Yeah, those are temporary ones.
Yeah,
they
you can do those.
Work for like a day.
Yeah, well, actually at ASTMH, they had those kinds of stick-ons of mosquitoes. You could put a mosquito tattoo.
Cool.
Temporary. Although a permanent mosquito tattoo would be cool. You know, a lot of biological things make great tattoos. Mm-hmm. Did I say I'm Vincent Racaniello? You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.